Hey everyone, welcome to The Explainer. I am absolutely thrilled you're here today. We are putting on our genetic detective hats and diving into literally one of the most foundational tools in modern biology. I mean, if you've ever wondered how scientists actually compare genetic codes in the real world, you are exactly in the right place. So let's jump right in and master the ultimate molecular comparison tool, NCBI's BLAST.
Okay, imagine this. You're at a microscopic crime scene. You swab a surface, run it through a sequencer, and boom, you are handed this exact string of text. ATGCGT, and so on, right? Just a raw, completely unlabeled string of DNA. Now, as a biological detective, looking at this raw data, well, it's kind of like looking at a fingerprint without having a database to check it against. It's this mysterious clue, but on its own, what does it actually mean?
Which brings us to the absolute core question we need to answer today. How do you identify an unknown sequence of DNA, RNA, or protein? How do you figure out if that random string of letters belongs to a human, some newly discovered virus, or, I don't know, a common houseplant? This is the exact problem that modern bioinformatics needs to solve. And thankfully, we have a digital magnifying glass that's perfectly designed for the job.
So here's our roadmap for this explainer. First, 1. Meet your molecular matchmaker. Then 2. Step-by-step BLAST tutorial. After that, 3. Decoding your BLAST results. And finally, 4. Real-world BLAST applications. Let's do it.
Section 1. Meet your molecular matchmaker. An introduction to the basics of BLAST.
Right, so the crucial starting point is understanding what this acronym actually stands for. BLAST is the Basic Local Alignment Search Tool. It's a massive bioinformatics program developed by the NCBI, the National Center for Biotechnology Information. And you really just want to think of it as the ultimate biological fingerprint database. It takes your unknown sequence and rapid fires it against a massive globally shared database of known sequences to find similarities. Literally, this is how we identify unknown genes, annotate genomes, and untangle those really complex evolutionary relationships.
But here's a really interesting part. BLAST isn't just one single tool. It's actually a whole suite of tools, and you choose one depending on the exact evidence you have. Think of this as your quick reference cheat sheet. So if you're starting with DNA or RNA, you're going to use BLAST-N to compare nucleotide sequences. Got proteins? You'll use BLAST-P. But what if you need to cross-reference? Well, BLAST-X actually converts your DNA into a protein sequence first and then checks the protein database. T-BLAST-N takes your protein query and compares it against a translated nucleotide database. And T-BLAST-X compares a translated nucleotide to another translated nucleotide. Basically, you just picked the right lens for the evidence in your hand.
Section 2. Step-by-step BLAST tutorial. Running your first genetic investigation.
Get those virtual evidence bags ready, because we're going procedural. Step 1. You just open up the NCBI BLAST website. Simple enough. Step 2. Based on that cheat sheet we just talked about, you choose your BLAST type. Let's say we have DNA, okay? So we'll select BLAST-N. Now, step 3 is absolutely crucial. You paste your sequence using the FASTA format. Remember that mysterious clue from earlier? That greater than symbol followed by a title with the actual sequence right on the next line? That's FASTA format. It's the standard, super clean way to package your genetic clues so the system knows exactly what it's looking at.
Moving on to the final setup steps, which are all about targeting your search. Step 4 is selecting your database. You might choose N for the standard nucleotide collection, maybe R for non-redundant proteins, or RefSeq if you want strictly curated high-quality sequences. Step 5 lets you adjust parameters, things like word size, scoring matrix, or filtering out low-complexity regions. But here is a pro tip for you. If you're a beginner, you can totally safely stick to the default settings here. Don't sweat it. And finally, step 6, click that BLAST button. Just keep in mind, your processing time is going to depend entirely on how long your sequence is and the massive size of the database it's scanning.
Section 3. Decoding your BLAST results. Reading the match report.
Okay, so once the search finishes, your screen is going to populate with a graphic summary of colored bars and a descriptions table. But to really read the detailed alignment section, you've got to know the lingo. In that section, you'll see your sequence stacked directly on top of the matching database sequence. The query is the evidence you brought in, your input. The subject is what the database found for you. Identity refers to the exact matching bases or amino acids between the two. And gaps, well, those refer to any insertions or deletions that had to be made to get the two sequences to align properly.
Now, if you take absolutely nothing else away from this tutorial, please memorize this rule. Lower e-value equals a more significant match. An e-value close to 0 is a highly reliable match.
Over in that descriptions table right next to the organism name and score, you are going to see the expect value, or e-value. Think of the e-value as basically the polygraph test for your genetic match. It tells you the likelihood that this match happened entirely by random chance. You want that number as close to 0 as humanly possible to prove your investigation was actually a success.
So once you verify a good e-value, then you look at the percentage identity. Let's use a concrete example to interpret this. If your query matches a database subject with a 99% identity match, wow, that is highly likely to be a direct hit. You'd probably conclude, this is a known Homo sapiens gene, right? But if your identity match is much lower, say hovering around 30 to 50%, it doesn't mean the tool failed, not at all. Instead, it likely indicates a distant evolutionary relationship. Congratulations, you just found a biological cousin.
Section 4. Real-world BLAST applications. Why this tool changes the world.
We've looked at the software interface, so now let's kind of zoom out to the macro level. Grasping this simple sequence matching process is unbelievably fundamental to modern science. The applications are really staggering. In medical research, scientists use BLAST every single day to identify disease-related genes and study dangerous pathogens. In biotechnology, it's literally the foundation for gene cloning and protein engineering. Evolutionary biologists rely on it to build phylogenetic trees and compare whole species. And in agriculture, researchers are using BLAST to identify crop genes and engineer crucial disease resistance to, protect our global food supply.
But as good genetic detectives, we do have to fairly assess our tools, right? What is fantastic about BLAST is that it's fast, it's highly accurate, it's widely accepted across all research, it has massive database coverage, and best of all, it's completely free online. But it does have its limitations. It's not built for very complex structural analysis. The output is only as good as what you put in, so it absolutely requires a good quality input sequence. And if your sequence has a lot of low complexity regions, like repeating patterns of the exact same letters, well, the tool might occasionally produce false positives.
So, to avoid those little pitfalls, I want to leave you with a rapid-fire list of expert tips to make sure your future genetic searches are perfectly clean. Ready? Number 1. Always use a clean FOSTA format. Number 2. Choose the exact correct BLAST type for your specific data. Number 3. Adjust your database appropriately. Don't search protein databases if you only need DNA matches. Number 4. Check that e-value carefully. Remember, closer to 0 is better. And finally, number 5. Always verify multiple matches to ensure you're getting a consensus, not just a single lucky hit.
BLAST is an undeniably powerful tool that essentially unlocked the field of modern genomics. And now, you know exactly how to use it. It is basically an open library of all cataloged life on Earth, just waiting for your queries. So, armed with BLAST, what unknown genetic mysteries will you uncover in the vast NCBI database? The evidence is out there waiting for a match. Thanks so much for joining me on The Explainer today, and keep exploring.